So let's say we have a piece of double-stranded DNA. And DNA is really a very, very long molecule, so we're going to put in arrows at the ends of all of our DNA strands to remember that. And when DNA is ready to get replicated, it's going to start bubbling out in many different spots. And each one of these places is called an origin of replication. And the origin of replication is simply a sequence of nucleotides that tells all of the enzymes involved in replication that, oh, this is where you should start working. Let's take a closer look at one of these origins of replication. And sometimes it's called a bubble of replication or a replication bubble. And let's label our replication bubble. So if this is the 5' end, this is going to be the 3' end. And DNA strands are anti-parallel to each other. They run in opposite directions. So it means that the bottom is going to be 5' there and 3' over here. And an enzyme is going to come along. This enzyme is DNA polymerase. And let's see what DNA polymerase is going to do. So we'll start with the bottom strand. DNA polymerase is going to lay down an RNA primer. And the purpose of this RNA primer is it acts as a piece of nucleic acid onto which DNA polymerase can then hook new nucleotides. So DNA polymerase will then add nucleotides in this direction. Let's look at our top strand. DNA polymerase lays down an RNA primer and then proceeds to add nucleotides in this direction. Let's look at the strands that are being synthesized. So DNA is synthesized always in the 5' to 3' direction. So we'll label our top strand. We always go from 5' to 3'. So synthesis is happening in the 5' to 3' direction, but if you look at the parental strands, the original strands of DNA, they're actually being read in the opposite direction. They're being read from 3' to 5'. So let's make this very clear. DNA is synthesized in the 5' to 3' direction. And again, that's referring to the strand that's being put together. But DNA is read, and when I say that, I'm referring to the parental or template strands. DNA is read in the 3' to 5' direction. And now let's take our origin of replication or replication bubble and let's cut it in half so that we can take a closer look at what's really happening. And I drew it here, except that instead of a curve, I drew it as a straight line, but it's really the same thing. And let's label our replication fork. This is a replication fork because, as you can see, it just looks like a fork. Let's label the strand. So let's say that this is 5'. That means this is going to be 3'. And of course, the other strand goes in the opposite direction. And let's pretend like we are a DNA polymerase. What are we going to do? Let's start with the bottom strand. We're going to lay down a primer. Remember, it's made up of RNA. And we're going to proceed to add nucleotides in this direction. And even though I'm drawing the nucleotides as separate units, remember, they're really being connected to each other. But I just want to, emphasize the fact that synthesis is happening, so I'm drawing them as separate units. But, try to remember that they're really connected to each other. And just to remember, again, synthesis goes from 5' to 3'. Let's look at our top strand. We put an RNA primer over here at the 5' end. Then we proceed to add nucleotides in this direction. And I'm going to label that strand as well. It goes from 5' to 3'. Let's analyze what's happening here. In the bottom strand, we added an RNA primer, and then we proceeded to add nucleotides in this direction. And when this part of the fork opens up, this part right here, we can just add more and more nucleotides. But look at the top strand. We added an RNA primer and then added nucleotides in this direction. And then when we want to kind of continue to replicate this part of the DNA, well, we can't. We kind of have to wait till the fork opens up with enough room for us to, put down a primer and then add nucleotides. So let's see what this replication fork looks like a few seconds later. So in our bottom strand, we had an RNA primer, and we kind of just added nucleotides in this direction. And as the fork opened, we just kept adding nucleotides. And in this strand, here's the first primer that we added. And then when the fork opened up with enough room, we lay down another primer and then proceed to add nucleotides in this direction. Let's see what this looks like a few seconds later. So again, in our bottom strand, we had only one Rna primer, and then we added a bunch of nucleotides. And in our top strand, that's our first primer. And then we added some nucleotides, and then we put in another primer, and then added nucleotides. Then we put in another primer, and we added nucleotides. And I think you get the point. And so in the bottom strand, synthesis was continuous, and we call that strand the leading strand. But in the top strand, synthesis was not continuous, and we call that strand the lagging strand because it's always kind of lagging behind. It's always waiting for the fork to open up with enough room so it can put down another primer and then add all the nucleotides. And these fragments have a name. They're actually named after the person who discovered them. They're called Okazaki fragments. And just to give you an idea of what's really going on over here, I'm drawing maybe 5 or 6, nucleotides per second, but in real life, this happens at a staggering pace. In fact, the bacteria E. coli, the DNA polymerase in E. coli, will string together 1,000 nucleotides in 1 second. So that's a really large amount. And we humans are a bit slower. We put together about 50 nucleotides per second. But still, these are really impressive numbers. You could just imagine DNA polymerase zipping down a piece of DNA and, stringing together 1,000 nucleotides in 1 second. That's a very, very impressive thing. Let's go to our next step. So we've finished synthesizing our strands of DNA, but we have all these RNA primers here, and we don't want RNA primers stuck in the middle of our DNA. So an enzyme, RNase, is going to come along and remove all these primers, and then a different enzyme will kind of fill in the gaps with DNA. And I just want to point out to you, if you remember before, I told you that really all these nucleotides are bonded together. I just, left it out because I was trying to show you, how it was just being synthesized. But remember, these are all connected to each other. But not only that, these nucleotides, and again, I didn't draw this in the previous diagrams, as they're being synthesized, they're also being connected to the corresponding nucleotides on the parental strand. So just bear in mind that these are all bonds, and, just remember that this is how it really is. And I'm not going to fill in the rest, but I think you get the point. And our last step is, an enzyme, DNA ligase, will come and string together all of the disparate or separate fragments of DNA so that we have one continuous piece of DNA. Here's our newly synthesized pairs of DNA. So we have 2 pairs of DNA, and each pair is going to have one parental strand. That's the original strand that we were using as a template, and one daughter strand.